Chapter 23 focuses on all of our microorganisms that fall into the eukaryote category. So our prokaryotes are all considered microorganisms, and then our smaller, generally non-multicellular organisms, or our single-celled eukaryotes, fall under the category of microorganisms. And then we also have our fungi. So fungi can be slightly larger, but they fall under the microorganism category because most of them are going to be small. And the majority of the organism for a fungus tends to be underground with very small hair-like structures being the main part of those organisms. So even though we can see some fungi, like these blue oyster mushrooms here, we sort of still consider them smaller organisms. They're certainly not large on the level of a giant sequoia or a humpback whale or something like that. So we're going to continue chapter 23 talking about the origins of multicellularity, which we already touched on a little bit in chapter 22, and then also talk about the characteristics of some of the fungi. We will go through a couple of the groups of fungi, but very, very surface level, kind of like we did with the protists, just very broad categories, and we're not even going to hit all of the fungi. So our oldest multicellular fossil was probably similar to a red algae. Now, algae fall into the Archaeoplastida group, which are the closest relatives to the land plants. Land plants are more closely related to red algae, but red algae and green algae are relatively closely related. So in a multicellular organism, you start with a single cell. Now, usually that's going to be the result of fertilization, although there are some organisms who are asexual even in the eukaryotic cell world. But we're going to start as a single cell, and that single cell is going to divide into daughter cells, and they remain attached to one another. And we would call this development. So those cells divide, and then in a more complex multicellular organism, those cells are going to diversify into different tissues. So there are two possible origins for multicellularity. And the first is that individual cells came together and began to associate in a colony, and then through natural selection, that colony became dependent on itself. So all of the organisms within that colony became dependent on one another and could no longer live separately. And then as evolution continues, they would become one single organism that are then produced by some sort of reproductive mechanism. And so even though we talk about many cells coming together to form a colony, that's the evolutionary pathway. But now we have a single cell that divides to produce those multicellular organisms. The other potential origin for multicellularity is that a single cell divided. We would call that fission in the bacterial world. Fission would have happened, and then instead of separating, they would stay stuck together and eventually evolved a mutual relationship and evolved into a single multicellular organism. So the mechanism doesn't matter too much. It may have been one way in our plant lineage. It may have been one way in our animal lineage. Doesn't matter too much because the end result is the same, that multicellularity evolved in 3 different lineages. So our early eukaryotes were still single-celled. The earliest widely accepted eukaryotic fossil dates to about 1.8 billion years ago. But that's just the point when a eukaryotic cell fossilized. Eukaryotic cells may have begun diversifying well before that, and at this point is just where we actually see the first record of this. So this indicates that there was some diversity by the time we actually see fossils. So that 1.8 billion years ago is just the earliest that we can verify. So as our evolutionary pathway continues, we start to see diversification in type. That very first one was red algae-like. By 1.5 billion years ago, we have another fossil that appears to be algae-like or fungus-like. And we start to see a little bit more variation compared to our prokaryotic cells. So, of course, they have a nucleus because they're eukaryotes. They have a flexible membrane. Remember that most of our prokaryotes have a cell wall, but not all of our eukaryotes have a cell wall. They also have a flexible cytoskeleton, which means their shape is a little bit more irregular. This is going to be the precursor for the ability to move. About 1.3 billion years ago, we begin to see the first evidence of a multicellular organism. Here we've got this Bangiomorpha, which is an ancient red algae. This guy had 3 different cell types, and this indicates our first direct evidence of multicellularity. So we've got eukaryotes about 1.8 billion years ago, and then multicellularity coming up about 1.3 billion years ago. But remember that multicellularity evolved in our plant line, our fungi line, and our animal line. So we've got multicellularity happening in 3 different places. All right, so as we move along, by about 635 million years ago, so we're getting much, much closer to present day here. I know it still sounds like a lot, but on the evolutionary time scale, we're getting much closer. We start to see larger and more complex eukaryotes. We also start to see more complex communities, and algae would be sort of the base of the community, just like plants are the base here, our primary producers, or I'm sorry, not here, now. And those algae would be consumed by larger organisms. And then we've got even larger, more complex ones arising after 600 million years or so. And then we finally start to see early animals. They would have had soft body parts, but were starting to get quite large. And those early animals begin to appear about 535 million years ago. But we are still talking about fungi, so we are going to continue our discussion on the microorganism eukaryotes, and then we'll move on to animals a little bit later. Our volvocine green algae are important because they appear in a variety of multicellular forms. So there are different species that have different levels of cell organization and numbers of cells in their multicellular form. So on the left, we have the Chlamydiamonas, I can never say this word, which is a unicellular eukaryotic algae. And then next to that, we've got the gonium, which is composed of 16 identical cells. And you can see they are arranged in a very specific arrangement. And these are considered a multicellular organism. So it is 16 cells in this organism. We wouldn't think that's very many, but it still is one. So none of them can live without the other. This is different from a colony. And then next to that, we have the Pleodorina californica. These are anywhere from 64 to 128 cells, and these guys have 2 distinctive cell types. And those cell types are going to be really important as we get into our higher eukaryotes, more complex eukaryotes. And in the Pleodorina, we have both the somatic cells and the reproductive cells. So somatic are our non-reproductive cells, and then we've got the reproductive cells as well. And then next to that, we've got the Volvox aureus. So we're moving up in complexity up to 2,000 cells here with, again, 2 different cell types. This Volvox is the type that we're going to look at in our lab. So our fungi have a few distinguishable characteristics, like all of our other groups. First of all, they lack plastids. The reason that I say that at the very beginning is that in a second I'm going to tell you that they have a cell wall. And sometimes students think, oh, cell wall, that must mean they're plants. And so the reason that I start off with saying that there's no plastids is so that you don't think that they are related to plants because of the wall. So lacking plastids means that it does not produce sugars by photosynthesis. So it is a heterotroph, it is not an autotroph, and it absorbs its food instead by digesting the living material, the organic material around it. Animals do the same thing, except they ingest their food instead of absorbing it from the outside. So most of our fungal species are going to have cell walls. Plant cell walls are made out of cellulose. Fungi cell walls are made out of a different polysaccharide called chitin. Chitin is also found in some animals. Our arthropods, for example, like our crayfish, our lobsters, our crabs, things like that, they all have chitin, and their chitin forms their outer shell. So not related to them very closely either, but they do share that similar characteristic. Some of our fungi are unicellular, which is why we put these guys into the microorganism category. Up at the top, we've got Saccharomyces cerevisiae. This is baker's yeast. So this is the type of yeast that's used for bread making and brewing. They are fermenting organisms, although they also can do cellular respiration. But that fermentation is how we get some of our food products. There are also multicellular fungi, and these fungi produce, or actually are made up of these microscopic threads called hyphae. So down here on the bottom, we have this organism called trichoderma, and trichoderma, along with many other fungi, use these hyphae. So this is an electron microscope image of these hyphae. So this is how tiny they are. This is only 4 micrometers right here. So this is very, very small, very, very thin. And the hyphae are important because they extend through the substrate that they are living on. And that is how they acquire their nutrients. So we'll talk about them a little bit more, and we'll talk about how a mushroom, even though I'm telling you they are made up of these microscopic hyphae, how we get a mushroom out of this structure. The mushroom is actually just a reproductive part. So we'll talk about that in just a minute. So just to sort of let you know where we are in the Opithecont supergroup, the fungi kingdom. Animals also fall into the Opithecont kingdom. We'll talk about them a little bit later after we wrap up our fungi and our plants. So our fungi, it should say contained within the Opithecont supergroup. The fungi kingdom diverged from the other Opithecons, the ones that would lead to the animal lineage, more than 1 billion years ago. So we say they're more closely related than plants, but they still diverged quite a while ago. They're a monophyletic group, which means all of the fungi organisms fall into the same evolutionary group. And there are more than 100,000 species of fungi, so there are a whole bunch of them. We did talk about some fungi-like eukaryotes in our protist section, and that just means that they have some characteristics that are similar to the fungi. It does not mean they are part of the fungi group. Like all of our lineages, they started out relatively simple and were mostly aquatic. But like our plants and animals, they did move on to land as well. And they actually became very good at living on land. And we will talk about how they helped plants, and therefore a little bit later animals, be able to move to land as well. So our fungi are heterotrophs, which means they have to ingest other organisms to get their organic molecules. And they use absorptive nutrition. So both animals and fungi do use absorptive nutrition. When they do this, they secrete digestive enzymes to break down the organic materials in the food source that they are eating. However, animals do this in their digestive tract, so in their intestines and in their stomach. They break down the food and then absorb the nutrients directly into their bloodstream where it is processed and all of that business. Fungi do this by secreting digestive enzymes outside of their hyphae, so released from their hyphae into the substrate around them. So this might be leaf litter, it might be a decaying log, it might be the orange that you leave on the counter. It can be whatever it is that they prefer for their food source. So once those digestive enzymes are released, they break down the organic material, and then the small organic molecules can be absorbed into the cell. Both animals and fungi store carbohydrates as glycogen. So plants store their carbohydrates as glucose, as starch. Both glycogen and starch are glucose, but plants store their energy as starch, if you think about a potato, and then animals and fungi store it as glycogen. It's just a slightly different form of our glucose polysaccharide. I already mentioned a little bit about absorptive nutrition, so I'm not going to say anything else here. If you would like to write down some notes, go ahead and pause the video and then start it back up when you're ready. The main structure of the fungal organism is called the hyphae, and these are individual long, thin, branched filaments that are made up of those fungal cells. So all of the hyphae together are called the mycelium, and we can see down here on the bottom a picture that shows the individual hyphae down here in the soil growing through. You can see how highly branched they are. And then this whole structure here is called the mycelium, and actually everything up into the top as well. But this is all mycelium. You can see in this image here all these white thread-like structures. Those are your hyphae. And this looks like it is sort of growing through some maybe like decayed branches or garden mulch, something like that. When the organism is getting ready to reproduce, it grows hyphae up into this reproductive structure. That reproductive structure is called the fruiting body, and they are special mycelia that are there to produce spores. But when they are just digesting their food, that mycelia is going to spread through the food. There were some examples on the previous slide of what that food source might be. This is where they secrete their digestive enzymes and then break down the material that they are attached to. When fungi reproduce, they reproduce by two or more hyphae from neighboring mycelia growing towards one another and fusing in the center. So here we've got the orange and the yellow hyphae meeting one another, and you can see that where they meet, they produce hyphae that grow up into this structure at the top, the fruiting body, and then those hyphae release spores. So those spores are then going to germinate into more hyphae somewhere else. On the right-hand side, we have a bunch of different examples of fruiting bodies. They come in all kinds of different sizes and shapes and forms. And they're really just here to hopefully you kind of recognize some of the structures. You might have seen some of these out hiking or whatnot, especially if you do truffle hunting. Like the top is a black truffle up there. On the right is a puffball fungus. It releases its spores in a poof. So you can see that cloud of spores up there on the top. So there are just a variety of ways to reproduce and release spores so that more hyphae can grow farther away.